All right. So we talked quickly about problem design, hypothesis, all of that kind of stuff when you're putting your lab report together. Please make sure you have a look at that. One thing I forgot to mention about the hypothesis, okay, we said that in the if part, okay, we were supposed to talk about what a reaction was. Okay, a good place to look for that, okay, for what a reaction is back on the first day. Okay, the very first day we talked about Dalton's atomic theory, and it had 4 points. Okay, and the third point talked about what Dalton said happens in a chemical reaction. So I would say refer back to that and try and integrate what he said about separating, joining, rearranging, all that kind of stuff. Okay, I think that would be a good thing to kind of work into the if part of your hypothesis when you're doing that. Okay, the and part talks about the experiment, then is what we're going to see. Okay, now for today, you're going to have your charts, okay, that you're going to have to fill in. Right, we're going to do the prediction part for the reactions before we go across. Okay, but we're going to need to have something to record before and after observations in, okay, so that we can fill in our observations chart on the Google Doc when you are putting your lab report together. Okay, so please make sure you take at least a blank piece of paper across at the very least so you've got something to write on. Okay, so for today, okay, when we get across to the lab, this is what you're going to see on the front table, okay, are all of the samples. Most of them are already in solution, okay, so they have little eyedroppers, okay, disposable eyedroppers in them. What do you need to make sure you do with those eyedroppers? Don't mix them, all right? If you take the eyedropper from one sample and use it in another, the reaction will happen in the sample, and I will be displeased because I will have to make the sample again, all right? And I don't want to have to do that. So if you forget which one it came from, just tell me, okay? And we'll throw that pipette away and we'll get another one, okay? It is better to throw those away, they're disposable anyway, okay, than it is to contaminate a sample because you can't remember where you got it from. Okay, so if that happens, just tell me, we'll throw it away, we'll get another one. That's way better. Okay, everybody clear on that one? Okay, so on the beaker, or sorry, on the Erlenmeyer flasks, okay, written in red permanent marker on the sides is the chemical formula for what is in there. All right, so for example, for sodium hydroxide, it's going to be NaOH. Okay, so you got to just look on your materials list. Okay, if you go back to here, on your materials list, the name and the formula is beside everything. So if it says you're mixing for like pair 1, A and B, okay, you need to look for NaOH and NH4Cl. Right? Now, the other thing we're doing differently, okay, because I listened to you, okay, you said one of the sources of error in the lab, for the first lab we did, was that everything was really, really small in that depression plate. So, we're not going to use the depression plates. We can't have a whole bunch of reactions occurring side by side in a little plate. If any of them bubble or foam, they'll contaminate all the others. So we're going to do one reaction at a time in a watch glass. Okay, watch glass is a piece of glass that's about this big. Okay, so that's a little bigger than actual size on the screen there. Looks like a big contact lens. Right, rolls around a little bit, so don't bump it. Okay, so it's just going to sit on your tabletop. And you're going to put the samples in there. Now, you don't want the reaction to happen right away. So you'll come up and put one of the samples. So if you're doing pair 1, let's say put A in the watch glass. Okay? Take the watch glass and an eyedropper full of B back to your station to mix them. Right? Because what else do you have to be measuring when you start the reaction? Temperature. All right, so you need to have it back there, have the thermometer in, mix the two, and start recording your observations. Is there a temperature change, precipitate form, change in odor, bubbles, all of those things we talked about? Okay, record anything that you see. Everybody follow? Okay, when that's done, okay, when each reaction is over, take the sample with the wash bottle and rinse it into the disgusting bucket. Okay, none of this stuff should go down the sink. All right, it is all very concentrated. Right, because I want the reactions to be obvious. All right, everybody with me there? Okay. Now, these reactions, they do not need to be done in order. Okay, with the exception of, you must do reaction 5 right after reaction 4, because if you read down here, okay, pair 5 is C and the product of pair 4. So if you do pair 4, mixing A and F, keep that. and then go get some of C and add it to what you have. Those are the only two you actually have to do 4 or 5. Okay? The rest you can do in any order. And I would suggest you try and pick some at random. Otherwise, everyone's going to be fighting over the same 2 samples at the same time. All right? And that's just not going to make efficient use of time. So a few of you can start at the end, a few of you start in the middle, a few groups start at the beginning, okay? And we shouldn't be fighting over the same samples too much. All right? Now, when you get in there, okay, please make sure if you have long hair that it's tied back. Okay? If you really like the clothes you're wearing, make sure you get a lab apron on, okay? Because the sodium hydroxide, the acetic acid, and the hydrochloric acid are super concentrated. If you get them on your clothes will have a hole probably before the end of class. Okay? So yeah, you don't want to be spilling any of these. Okay? Also make sure your eye goggles are on and that they stay on. They are not forehead protectors. Okay? Guys, it is not fun for me to have to put somebody's face in the eye wash station. Okay? The last time I had to do that, both the person in the eye wash station and me had bloody noses. Okay? I kid you not. Right? They were flailing like crazy because they had really bad stuff in their eye. You imagine what it's like, okay? Your eye is burning, okay? And they smoked me right across the face. And then I grabbed their head and shoved it into the eye wash station and bashed their bridge of their nose right against the center of the eye wash station because your adrenaline gets going, okay? So, yeah, let's not have that happen, okay? Keep your goggles on. That's why you're given the goggles. Nobody gets hurt if we wear the goggles, okay? Now, other things here to be aware of. Remember, you're rinsing after each one, rinsing with the distilled water. Okay, if your wash bottle's empty, just fill it from the big container there at the back sink. Other than that, I think we should be, the phenolphthalein, that's this stuff here, the 0.1 mole per liter phenolphthalein indicator. That's the only thing that's not in an Erlenmeyer flask. There are 4 little plastic squeeze bottles, okay, that are labeled phenolphthalein. Okay, this is the stuff they use in CSI to test for blood. Okay, so they swab with the Q-tip and then they put that stuff on, turns pink. Okay, that's the same stuff. We're using that today, okay, as one of our reactants. So you just need a few drops of that. Let's say 3. Okay, so when you're using the phenolphthalein, how many drops do you need? 3. Okay. Okay, let's do our prediction stuff here. Okay, so you'll need a sheet of paper. Okay, we're going to look at predicting these chemical reactions before we go across the hall. Okay. Is it possible that when we predict some of these reactions right now that we might be wrong? It is. Okay, it's entirely possible. That's why we want to record our data. We may have to revise our predictions a little bit when we do our actual report. Now, these predictions need to appear in your lab report. Okay? They need to be like kind of right before the procedure as planning. Okay? We want to compare our predictions to what actually happens. All right, so pair 1, I've got sodium hydroxide being mixed with ammonium chloride. Are those both ionic compounds? I don't know. Are they? Yes, they are. Okay? Ammonium is a polyatomic ion acting as a metal. Chlorine's a nonmetal. Metal, polyatomic ion. It's got to be ionic. Okay? So, what kind of reaction starts out with 2 ionic compounds? Double replacement. Okay, what do we do in a double replacement reaction? Switch the metals. Remember, ammonium is acting as a metal in this reaction. Okay? So, you guys can predict, okay, what you think the products are going to be for that reaction. Okay, switch the metals and you'll have your products. NH4Cl. Okay, for pair number 2. Right? I've got acetic acid. Okay? Now, it's written funny because it's a weak acid. Okay? Hydrogen's acting as the metal in this one, even though it's listed second. Okay? But when you have an organic compound, you always have to write it with the carbon first. It's weird. Okay? So this is really, okay, H plus and CH3COO minus. Okay, that's how it looks. So is it an ionic compound? It is. Okay, this is an ionic compound. Hydrogen is the metal. Okay, this stuff, is it also ionic? Is sodium a metal? Okay, and this is a polyatomic ion, hydrogen carbonate. Okay, so we've got sodium, hydrogen carbonate. So what kind of reaction are we looking at here? Double replacement. A lot of these are double replacement. Okay, so we know hydrogen's acting as a metal, sodium's acting as a metal. In a double replacement reaction, we switch the metals. Okay, so you should be able to figure out who ends up with who on that one. Okay, for pair number 3, are they both ionic compounds? Okay, is it another double replacement reaction? Okay, so we got to swap the metals on that one. Okay, do your predictions, okay, with the metals swapped there. Okay, and then we'll look at 6 and 7. Okay, looking at pair 6, I've got potassium iodide reacting with lead 2 nitrate. Okay, are those both ionic compounds? Okay, is this another double replacement reaction? It is. Okay, so if they're both ionic compounds, we got to swap the metals. Potassium's a metal and lead's a metal. Okay, so we know who the partners are on the other side. Don't worry about balancing it right now. You can balance it later. We just want to kind of have the products predicted a little bit. Make sure you swap and drop the ionic compounds that you make on the other side. Okay, because for all of these so far, we've made ionic compounds on the other side that will need to be dropped and swapped. Okay, this is lead 2. Okay, so it has a charge of positive 2 for when you do that drop and swap. Okay, and for pair number 7, I've got zinc metal element, okay, reacting with hydrogen chloride. What kind of compound is that? Okay, it's a single replacement reaction because this is an ionic compound. Okay, is hydrogen acting as a metal or a non-metal? Metal, it's listed first. Okay, so we got a metal reacting with an ionic compound. You guys can figure out who needs to switch with who. Okay, predict your products on the other side, and then we're going to head across the hall. All right, a couple of things here, guys, in terms of stuff that needs to happen for your analysis. Okay, for question number 1, using words and formulae. So that means write the equation both ways. Write out all the names and write out all the formulas, balanced. Okay, write the chemical equation of each chemical reaction, obviously except for pair 4 and 5, because we have not done acid-base indicator reactions. It's not a type that we do in Science 10, so you do not have to do 4 and 5, but you do have to do 1, 2, 3, 6, and 7. Now, I think you would agree that in some of our observations, our predictions don't agree with what happened. All right, for example, in this pair, pair number 2, we mixed vinegar with baking soda. That's what's going on here. Acetic acid and sodium hydrogen carbonate, vinegar and baking soda. Okay? We predicted 2 ionic compounds. What happened in this reaction? A gas was formed. Can ionic compounds be gases? They cannot. All right, so something else happened in this reaction. Right? We have to figure out what when we write the reaction out. Okay? I will give you one little tip. The gas that was formed in this reaction is carbon dioxide. All right, now that carbon dioxide came from here. Okay, so what's left after we've accounted for this carbon dioxide? What's left is sodium with this hydrogen and this oxygen. Okay, that's still left. Okay, and all of this stuff is still left. So what we've got to figure out now is who ends up with who. Okay, think about what's positively charged and what's negatively charged here. Okay, and see if you can figure out who might end up with who now as a replacement reaction that we've accounted for the carbon dioxide. Everybody with me? Okay, maybe if I write this way, it might make a little bit more sense. Okay, what about pair 1? What happened in pair 1? Yeah, should have been a really strong smell, which means what was formed? A gas. Okay, did our prediction indicate a gas? It did not. Okay, as a little hint, the gas that was formed is ammonia, NH3. Okay, that ammonia came from here. Right? So what's left in this compound after that? Okay, 1 hydrogen and 1 chlorine are left. You got to figure out what the other products of that reaction are now. Hey, everybody with me there? Okay. The other ones, did they work out pretty much like we predicted? Did it seem like we got ionic compounds or ionic compounds and solids or whatever when we were done? Okay. Now, what we got to look at here is one of the questions also asks you, 2 reactions produce precipitates. At least they should have. Okay. 2 reactions should have produced precipitates. One of them was pretty obvious, right? The yellow stuff. Okay. And one of them should have produced what color precipitate? White. You should have got a white precipitate in one of the reactions. Okay, you better figure out which one. Okay, so what you need to do for analysis question number 2. Okay, for analysis question number 2, it says, okay, use the chemical equations you wrote and your solubility chart. So when you predict the products, you've got to check from your solubility chart which product was the precipitate in that reaction. Okay, everyone follow me there? You guys got to do that for that part. Okay, then for your conclusion, copy and paste your hypothesis. Tell me, based on your data, was it right or wrong? Did we see those signs of a chemical reaction? Did we see every sign we predicted? Doesn't have to be in every reaction. Did we at least see it once? Okay, that kind of stuff. Then your sources of error. Okay, how many do you have to give me? 2 sources of error with ways to fix them. Okay? You got to include that. All right, the class discussion you don't have to do. We'll talk about that when I return your labs on March the 10th. Okay? All right. That should cover it all, guys. You should be able to get what you need from there. If you missed today or you lost your data or you're not sure about your data, okay, I included the lab data in Google Classroom. Okay, there's a PDF file that included the lab data right in the reactions assignment. Okay, so if you go in there, one of them will be reactions lab data, and you can double check. Okay, the reactions lab data looks like this. Okay, so you can just double check yours with mine and make sure that you got about the same stuff there. Okay. Everybody with me? Okay, you've got about another 10 minutes or so. Okay, maybe sit down with your group, start figuring out what those reactions should be while you can put a few heads together. Okay, stuff like that. But we are working until the bell. So that means everybody's books are open and they're accomplishing something right now. Okay? Remember, this is due March the 9th, guys.